Welcome back. And as we continue through Chapter 9, our next big topic is looking at sort of inheritance patterns that are more complex than Mendel's patterns that he picked. Mendel intentionally picked traits that were very simple traits to follow, like purple and white flower color or wrinkled and round peas, because he knew he was starting from scratch. Certainly in the 150 years since Mendel, though, we know not all traits are quite that simple. Last time we spent an awful lot of time talking about what we would call a Mendelian trait, a trait that follows Mendel's basic laws and basic ideas, his laws of dominance, segregation, and independent assortment, a trait in which there's one gene that largely determines phenotype. And there's a dominant and recessive allele, and those are going to combine to give us that phenotype, and the environment tends to play a very small role in determining phenotype with a Mendelian trait. As I just mentioned, much has been learned since Mendel, obviously in 150 plus years. Now we know his basic ideas hold true, that we've got two alleles for trait in each adult. We've got one set of DNA again from mom, one set from dad, and that when we make our gametes, that law of segregation tells us we're going to put one of those two versions of that gene into either an egg or a sperm cell. But a couple of areas where we do see some variation in Mendel's ideas, one in particular is the expression of the alleles. Again, genotype getting turned into phenotype is expression here, and so that's not always quite as simple as what Mendel explained. So we're just going to go through today and look at 5 different exceptions to Mendel's basic ideas. Now, this is by no means an exhaustive list. There are many, many more exceptions, but these are 5 of the more common exceptions we see in terms of different traits and their inheritance patterns. Before we jump into the traits that we're going to look at in these different exceptions, I want to mention this idea of superscripts. Now, we talked about last time with Mendel that we're going to use a letter to represent a gene, whether it's P for flower color or T for tongue curling, we can pick that letter. Well, we can only use the capital and lowercase letters like we did last time if we have a Mendelian trait, if we know that one allele is dominant and will cover up the other and be recessive. But some of our examples today, that's not going to be the case. We're not going to see a dominant recessive. We may see both genes sort of being equal and being both expressed into the phenotype. And so if that's the case, we're still going to pick a letter to represent our gene, but we cannot use capital and lowercase. Instead, to tell alleles apart, we're going to use superscripts to be able to tell our allele. So for example, if I was looking at flower color, I might call that the C gene for color. And if I've got two different alleles, a red and a white allele, again, I can't call one big C and little c unless I know one's dominant and recessive. So instead, I'm going to call one C with a superscript R, tell me that's the red color allele, and the other one C with a W, meaning it's the white allele. And so we're going to stick with one letter, but we're going to use superscripts to tell the different versions of the gene apart. Don't let this fool you. This is just a way to write down the different alleles, so don't let that extra superscript bother you. One example of these exceptions beyond Mendelian traits is what's called incomplete dominance. In incomplete dominance, both of the alleles are equal, meaning if we have a heterozygous, both are going to be expressed. We will not have one dominant and one recessive. We will not have one covering up the other. And a good example of this is looking at flower color in certain plant species. For example, carnations are a common example, or common snapdragons. And these are going to use the system we just mentioned on the last slide. There's a color gene, there's a red, and there's a white allele. And so we'll call that C superscript R for red, C superscript W for white. Okay, and again, we can't use big C and little c or big R and little r if we don't have dominant and recessive, which in this case we certainly do not have. So if we look at this incomplete dominance in flower color, certainly some plants could get 2 copies of the red allele. So they would be homozygous for red, and obviously that would produce a plant with a red flower. Likewise, if one was homozygous for the white allele, that would be a plant that produces a white flower. Where this is going to differ from Mendelian genetics then is what happens if I cross those two, if I were to cross a red with a white. Now the red plant is always going to pass on red information, a red allele, its gametes. The white always going to pass on a white allele in its gametes. So when we do our Punnett square, it's pretty simple, and we're going to get the whole next generation are going to be heterozygous, one red and one white. And in this case, since it's incomplete dominance, both red and white are going to express. Oftentimes they blend together. So in this case, red and white are going to make pink. And so in the case of something like a carnation, if we cross a red and a white carnation together, all the offspring are going to be pink. And here we can see carnations. And incomplete dominance in a way is simpler even than what Mendel described, because if I know the phenotype, I can figure out the genotype, because there's only one way to get a red flower, to have 2 copies of the red allele. Only one way to get a white flower, 2 copies of that white allele. And then of course if I have a pink flower, it must be heterozygous, 1 copy of each. And so our basic genetics hasn't changed in terms of how we figure out the gametes and do the Punnett square. That's all the same, but what's different is the expression. In this case, we're not going to see red cover up white or white cover up red. Instead, they blend together and they make pink. A good example of a human trait that follows this incomplete dominance is what's called hypercholesterolemia, which is certainly a mouthful to say, but if we break that down, hyper means more or high levels. Cholesterol, obviously, is certainly going to be a problem associated with heart disease, and emia, anything that ends in an emia, that suffix, has to do with your blood. And so basically, this is having high cholesterol levels in your blood, certainly one of the leading causes of increased risk of heart disease, stroke, heart attack, high blood pressure, and so on. And it turns out this hypercholesterolemia is one of the leading factors in terms of inheritance patterns where we see heart disease may potentially run in a family. And this cholesterol in your blood comes from LDLs, or these low-density lipoproteins that we might get in our diet, basically things like saturated fats. These LDLs are absorbed in our blood by our red blood cells, and they're absorbed through receptor-mediated endocytosis. And so kind of a ghost haunting us from the past here. Remember, with receptor-mediated endocytosis, we've got protein receptors on our blood cells. They're going to bind this LDL, and then they're going to take it in through endocytosis. And once that LDL is inside the blood cell, it's pretty harmless. Your body can break it down. It's not going to end up as cholesterol. The only worry here is if these LDLs remain in your blood plasma, then they're going to lead to higher cholesterol levels. And so these protein receptors are our way to remove the LDLs and keep our cholesterol levels lower. The inheritance issue associated with hypercholesterolemia then is looking at the number of these protein receptors. It turns out that there's two different alleles. We'll call them H for hypercholesterolemia. HA is the wild type or the normal healthy allele that results in producing these protein receptors that can bind the LDLs and remove them from your blood. But there's also an HI allele. We'll call that I for inability, which results in no proteins. And so it's a mutation where you don't make any of these receptor proteins. Well, in the case of being homozygous for type A, you're going to produce lots of receptor proteins. Your blood's going to be very efficient at removing LDLs. You're probably not going to have a cholesterol problem unless you really have high amounts of saturated fats in your diet. At the other end of the scale, a person that has homozygous for that HI, that inability, will have no protein receptors on their blood. Any fats that they take in will remain in their blood and become cholesterol. And we're going to see very high rates of heart disease in an individual like this. This would be a very severe form of the disease. Our incomplete dominance comes in between there if you're heterozygous. If you've got a copy of A and a copy of I, that means you're going to make a few protein receptors. Again, sort of red and white make pink, a lot, and none make a few if you're in between. And this group in the middle is where diet and exercise can really play a big role, because if you've got a few protein receptors, if you have a low-fat diet, your cells have enough receptors to remove the fat, and you're not going to have a big cholesterol problem. But if you're having a lot of fat in your diet, your cells are not very efficient at removing the fat. Some of those LDLs stay in your blood plasma, and we're going to see high cholesterol levels. And so this is a direct result of this incomplete dominance inheritance pattern. Another exception to Mendel's basic idea is what we would call multiple alleles. And Mendel described there are always two alleles, purple and white, or round and wrinkled. And again, this is because he was picking relatively simple traits. But as we learn more about DNA, we're finding that for many genes, there's more than two alleles, that there may be 3, 4, or even more alleles or different versions of a particular gene. Now, we need to keep in mind that as an individual, we each only have two alleles. And so I'm going to have two alleles, you'll have two alleles, but if I were to look at a group of maybe 30 or 40 people, there might be 4 or 5 different versions of that gene, or 4 or 5 different alleles that we might see in everybody's genotype. And a really nice example of this multiple alleles that deals with humans is our blood type, the ABO blood group. Whether you're type O blood or AB or so on, that's a genetic trait. It's due to a single gene, this ABO gene, we'll call it, and this gene has multiple alleles. In particular, there are 3. There's the A allele, and we'll write that I superscript A, and the I just stands for immunoglobulin, which is a big fancy scientific word meaning it's going to be a protein that we stick on our blood cells. And so the A allele codes for a type A protein that ends up stuck on the outside of our blood cells. There's also a B allele, we'll write that I superscript B. The B allele codes for a type B protein, a slightly different protein that ends up on our blood cells. And then finally, there is the O allele. And we're going to write the O allele as a little I because it's recessive to the other two. It gets covered up by those other two. And in particular, the O allele results in no protein. You don't make A or B's if you have this O allele. And so these are the three alleles, A, B, and O. Each of us has two alleles, and depending on which two we have from that combination, that's going to determine our blood type. Let's take a look then at our blood type and blood inheritance here. On our chart here across the top, we've got phenotype, which is basically your blood type. We've got genotype, which alleles might give you that particular phenotype. How does your phenotype or blood type affect your blood cells? There's antigens, which are basically the proteins that we're going to make for our blood cells. And then finally, there's antibodies that we're going to produce in our plasma. And so your blood type is a combination of these things, not just your cells, but the antigens and antibodies that you have in your blood as well. So if you're a type A blood type, and type A is the most common here in North America, here in the United States. If you're type A, there's two ways we could have type A in terms of your genotype. Obviously, if you're homozygous for the A allele, you're going to be type A blood. But because that O is recessive, if you have an A and an O allele, you're also going to be type A. And so if you're A blood, we're not quite sure. You could be either of those two genotypes. What that means in terms of your cells is you'll have lots of proteins on your blood cells, but in particular, you will have this type A protein on your blood cells as well. So you're producing this type A protein or A antigen. And in addition to that, our blood plasma would recognize B as being a foreign protein. So in addition to having A proteins on our blood cells, part of the type A phenotype is having anti-B antibodies. And this is why we have to match up blood types. Because if you're blood type A and I give you some B blood, your body will recognize those B proteins, your antibodies will attack it and start causing your blood to actually clump or clot in your arteries and veins. Obviously, pretty serious health issue there. Type B blood works basically the same way, except with the B alleles and the B proteins. And so you could be homozygous for the B allele, or you could have a B and an O allele to be type B blood. Type B blood means you have the B protein stuck all over your blood cells, and it means you're going to recognize A as foreign, and so you would produce anti-A antibodies within your blood. Next up is type O. For type O blood, there's only one way to get that. Since it's a recessive allele, you have to have two copies of the O allele. And O is the second most common, by the way. B and AB are pretty rare, but A and O are pretty common here in North America. If you're type O blood, that means you don't make either A's or B's. And so in regard to this particular type of protein, your blood cell would be blank. Okay? And for recognizing things as foreign, you would recognize both A and B as foreign because you don't make either one. And so we would have anti-A and anti-B antibodies in your plasma. Our last type then is type AB. There's only one combination of alleles left, and that's if you have an A and a B allele, and that's going to result in both proteins on your blood type, A's and B's. And this is another example of incomplete dominance as well. Our heterozygous ends up showing both of the characteristics. So type AB blood, you're going to produce both of those A and B proteins, and in terms of antibodies, you will not produce either one, and you better not, because if you did, you would recognize your own blood cells as being foreign. Okay? So this is our blood type inheritance. You may be aware that the universal donor, a person who can give blood to anybody in a pinch, is someone that's type O, because O blood does not have either A or B's on it. So if I was type B blood and I got some O, I'm not going to recognize it as foreign. And likewise, type AB is known as a universal recipient in terms of blood types. Since they recognize A and B's as being their own, they could take any blood type in a pinch. Okay? And we're certainly going to take a look in lab at some blood types, and we'll do a little bit of blood typing. Just as a side note, as we start thinking about blood types, because again, we will take a look at this in lab, you may be wondering, well, what about the plus minus if I'm A positive or O negative? And that plus minus has to do with a completely different gene, a completely unrelated gene called the Rh factor, or maybe we'll just call it the R gene. Okay? This really has nothing to do with your ABO blood type, but when we're matching blood, we need to match the ABO blood type and this plus minus or Rh factor blood type. And if we don't, then we might have a serious complication. So the reason we're familiar with the positive negative is we need to match it up in a transfusion, for example. Now this Rh factor gene is basically a Mendelian trait, a Mendelian gene. There's two alleles. A big R, or dominant allele, results in producing this Rh protein on your blood cell, and so that's kind of the positive allele for making that protein. There's a recessive allele, we'll call it little r, which results in not making the protein and not putting it on your blood. So if you're a positive blood type, you could have two copies of big R or just one copy of it. If you're negative, you know your genes, you must have 2 copies of little r. So for example, if I was type A positive, that means my blood has A proteins on it and this Rh protein on it both. Versus if I was a B negative blood type, that means I've got B proteins on it, but I'm negative, so I don't have any Rh proteins on it. And an issue associated with Rh is what's called Rh incompatibility. And you may be familiar with this, maybe you've heard of this before, especially for women who have had children before, you might have heard of this. Rh incompatibility is a case where if mom is a negative blood type and the father is a positive blood type and passes that big R on to the offspring. Because if that happens, mom's immune system will recognize that Rh as being foreign because her body doesn't make that normally. And so if the embryo develops a positive blood type and that blood comes in contact with mom's blood, then mom's immune system will start to attack that embryo and may reject that embryo and not carry it full term because of that immune system reaction. So that's called Rh incompatibility. It's usually not a problem with the first pregnancy because mom and the developing embryo don't swap much blood, at least until delivery. But once that happens in delivery and mom's immune system builds up antibodies to that Rh positive protein, then the second pregnancy or third might be at high risk of being rejected. So oftentimes if mom is a negative blood type, they'll test dad. If he's a positive, then they may recommend putting mom on some immunosuppressant drugs to try to reduce the likelihood of rejecting that developing embryo. Our third example of something beyond Mendel is what's called pleiotropy. And it's kind of an odd spelling, an odd looking word, but it is pronounced pleiotropy. And pleiotropy is when we have a single gene that affects more than one trait. So far, the P gene affected flower color or the ABO gene affected blood type. Well, in some cases, one gene, if it plays a central role in a particular organism, may affect many traits or many aspects of the phenotype. And a good human example of that is if we look at the hemoglobin gene. And we know that hemoglobin is a protein made up of 4 different polypeptide chains or 4 long chains of amino acids. They all fold together to make that functional hemoglobin protein. And within our cells, hemoglobin carries oxygen to the cells. It also gets rid of waste products like carbon dioxide. And there are 2 different forms of this hemoglobin gene, or 2 different alleles. The healthy allele we'll write as HbA for just hemoglobin type A, since it was the first one described. That results in the nice, normal, healthy hemoglobin. It's kind of a square shape when those different chains fold together, good at carrying oxygen and carbon dioxide. But there's a mutated form of the allele called the sickle cell allele, so we'll write that as HbS for hemoglobin sickle cell, that results in a slightly different sequence of amino acids, that the protein, instead of folding in this nice normal square shape, tends to fold in a long sort of thin shape. And certainly if we look at humans, sickle cell anemia caused by this sickle cell allele is by far the most common inherited disease or disorder that we see. So let's take a look at this hemoglobin type A and S and how they're going to impact not only sickle cell anemia, but another completely unrelated disease, malaria. Let's look at a quick chart then. Let's look at the genotype of which genes you have for these different type A and type S's, your blood phenotype, or whether or not you're going to have sickle cell anemia, and then this, again, our pleiotropic effect, a completely unrelated disease like malaria. And so you could be homozygous for type A, you could be heterozygous or homozygous for that sickle cell allele. If you're homozygous for type A, you produce nice, normal, round blood cells, and we've seen those in lab along the way, but here's a picture, nice and round, they're kind of donut shaped. Again, because that hemoglobin's square, it fits in those round blood cells. That's very important for those cells to be round because they bounce around and they get pushed around through your arteries and veins and capillaries. Someone who's heterozygous also is going to produce normal-looking blood cells, nice and round. But if you have 2 copies of that sickle cell allele, you will have sickle cell anemia. We know historically in humans and in most of the world today even that's fatal. It's unusual to see kids live past the age of about 4 or 5 years old with sickle cell anemia. However, in Western countries like the United States, we can diagnose sickle cell anemia at birth or even prenatally with a genetic screen. And if there's very aggressive treatment and it starts early and continues, we are seeing life expectancy of individuals with sickle cell anemia gradually creep into the 20s and even a little bit into the late 20s or even early 30s here. So in the last decade, a lot of progress has been made for treating sickle cell anemia. And here you can see what sickle cells look like. Because that hemoglobin stays long and skinny instead of folding up short, what it does is causes the blood cells to stretch out to hold that long, thin protein. These cells become long and pointy and sharp, and as they travel through your capillaries, they tend to get clotted and lodged, and that's going to cause a lot of circulatory problems. So that's certainly one aspect of the hemoglobin gene, is round versus sickle cells. But in terms of pleiotropy, your sickle cell genotype here is also going to impact your susceptibility to malaria. Now, malaria has nothing to do with sickle cell. Instead of being a genetic disease, we know malaria is an infectious disease. It's something you can catch. And most of us, when we think of malaria, probably think of mosquitoes. And while mosquitoes don't cause malaria, they help spread malaria. And so malaria is a problem in places where there's lots of mosquitoes, which tend to be warm, wet places like the tropics, whether it's in Central Africa or Central America or even Central Asia historically. We've seen lots of malaria problems. It turns out malaria is due to a type of bacterial infection called a plasmodial bacteria. And this plasmodium will get into your blood and it will attack and kill your blood cells. And we know that historically malaria has been fatal. Today we can treat it with antibiotics, but certainly a very serious disease. Well, it turns out that if your genotype is type A, you're homozygous for that wild type or healthy hemoglobin, you're going to be very susceptible to malaria. Meaning if you get bitten by a mosquito that has that bacteria in it, you're going to get malaria, and unless you get treatment, you're going to have some serious health issues. But individuals that are heterozygous, type A, type S, tend to be resistant to malaria, meaning if they get bitten by a mosquito with that plasmodium bacteria, they may get flu-like symptoms for a few days, maybe a little nausea, tired, fever, but then they're going to recover and they don't need that treatment, and so they have a resistance to that. And so we see that this hemoglobin affects both of these different diseases. And so because these two diseases are affected differently, we're going to see what's called heterozygous advantage. Now normally, if we have an allele that's fatal, like this hemoglobin type S allele, if you get two copies, individuals historically, at least in human populations, did not survive. And these fatal alleles typically are extremely rare in populations because, again, people that carry them have a good chance of passing it to their offspring, and their offspring don't survive, and so we don't see them lasting too long in the population. So having said that, though, if we look in certain Central African populations, we see that up to 60% of the people in these populations may be carriers, meaning they're heterozygous, type A and type S together. And again, this is off the charts high. No other genetic disorder is even 6% carriers would be high, but 60% is again off the charts, much higher than any other genetic disorder. The reason for this is this interaction with malaria. In Central Africa, it's no accident that malaria is a real problem. And so the cost of being a carrier, being heterozygous, is that if you get together with another carrier, 1 out of 4 of your offspring aren't going to survive. They're going to get 2 copies of that S allele and they're going to die historically. But the advantage in a place where there's lots of malaria, there's lots of mosquitoes to spread that malaria, is that if you're heterozygous, you have a better chance to survive to reproduce at all. And if you have 2 copies of type A, you may not even make it. And so in this case, where malaria is a real problem, the advantage of malaria resistance outweighs the disadvantage of that fatal allele. And so we see it becoming very, very common. Now here in the U.S., most African Americans can trace their ancestry back to these Central African populations. Now today we don't have a big malaria problem here in the U.S., and so the advantage of malaria resistance is gone, but the cost of that fatal allele is still there. So not surprisingly, we're seeing the percentage of carriers go down and down and down. Okay? Now anyone can be a carrier or have sickle cell anemia, but if you're African American, you're going to be in a high risk group, meaning you're more likely to have it than someone else. Everything else kind of being equal there. And so 10% is still a high number, but we are seeing it gradually go down. And this is probably one of the most commonly screened genetic disorders because it is so common. There certainly is a test available, and you can find out if you're a carrier and if you risk passing that allele on to an offspring. Next on our list is polygenic inheritance. And as the name implies, poly for many and genic, so many genes. This is going to be a case where a single trait or single aspect of your phenotype is going to be controlled by more than one gene. And so it may take 2 or more genes all kind of combining or working together to give us the phenotype that we're looking for. The simplest case would be 2 genes. We may see more than that. And a really nice example of a polygenic inheritance of 2 genes working together is if we look at skin pigment, in particular type of snake called a corn snake, which is a common snake throughout the Midwest, the very western part of Missouri, and then throughout Kansas and a lot of the central plains we see corn snakes. Corn snakes have 2 main colors or pigments in their skin. One is orange, so we'll just call that the O for the orange gene. And this is a Mendelian trait. It has two alleles, a dominant and a recessive. The big O, or dominant allele, codes for an enzyme that allows the snake's cells to metabolize or generate this orange pigment that ends up in the skin. But there's a recessive, a little o allele, which is a mutant form of this protein, that it doesn't work anymore. And so if a snake has two copies of this little o allele, it cannot make any orange pigment. The second color is black, so we'll call that the B for the black gene in these snakes. Works the same way. There's a dominant or big B allele that results in enzymes that can generate the black pigment for the snake, but there's a recessive or little b mutant allele that results in non-functioning enzymes. So if a snake has two copies of little b, it cannot make the black. And since both of these colors are involved in the skin pigment of these snakes, we have to keep track of both genes to figure out phenotypes, or if we were to do any crosses, to predict what their offspring might contain. So if we take a look at some corn snakes, here we have a wild type, or the most common form of a corn snake. It has both oranges and blacks in its skin. And so if we think of its genes, it must have a big O and a big B. Again, it could be big O or big O little o, the same for the B. But we know it has at least one dominant orange and one dominant black. Here we see a corn snake that has the black pigment, but no orange. And so we know that it must have two copies of little o and then a copy of big B to make that black pigment. We may on rare occasions see an orange corn snake, which certainly is very odd looking. It's making orange, so it must have a big O, but notice there's no B or black in it, so it must have two copies of the little B allele. And then finally, if we had a snake that was recessive for all of those alleles, it would be an albino. And albinos oftentimes look pink because the blood shows through the skin and gives it that pink hue. But in this case, that would be all the recessives, little O's and little B's. And so with our polygenic inheritance, we're going to have to keep track of more than one gene as we look at phenotypes and as we do things like Punnett squares and we do crosses. Many human traits also have this polygenic inheritance pattern, although for most of these human traits we don't know all the genes involved and we certainly don't know all the alleles yet. And it seems kind of odd that we would know more about a corn snake, for example, than we would a human. But remember, with different animal or plant models, we can do test crosses. We can cross different corn snakes together and see what they pass on. Can't really do that with people, and so a lot of times our knowledge of humans is a little bit slower than what we would like because of kind of our ethical issues in terms of experiments and our limitations. But we do have a few examples that I want to mention here. One is eye color. And eye color, your book looks at the example of what if there were two different genes, an A and a B gene, that would describe lots of different eye colors. It may even be more than that. In fact, it probably is. One of the things that we see with these polygenic traits is we start to lose our categories. Now, as humans, we like to categorize things. That helps us understand and remember things. And certainly, we like to categorize ourselves just like we do anything else. And so we might say someone has blue eyes or green or brown or black. In reality, our eye colors are a whole continuum from very light colored eyes like blue through the greens and hazels and browns and to the very dark colored eyes like black. And if we went out and got 100 people and lined us up by our eye color from light to dark, it'd be really hard to say, okay, here's where this color stops and the next color starts, because they all kind of run or blend together. And this is a common pattern with polygenic traits. We see this continuous variation. We start to lose our categories. The reality here is there probably are at least 3 genes with eye color. The color in your eye is determined by a pigment called melanin. And melanin is a brown pigment. It's kind of all-purpose in humans. We see more melanin in your skin makes your skin darker. More melanin in your hair makes your hair darker. If you put melanin in little circles, that makes freckles. More melanin in the iris of your eye makes your eye darker. And so someone with blue eyes has a very little melanin. Someone with green has more melanin. Someone with black eyes has even more melanin. And so that's certainly one important gene, making melanin. But there are probably other genes that determine, well, how much melanin do you make? Obviously, all of us make different amounts. And then maybe another gene that tells your cells, where do you put it? Where do you store it? Am I going to store it in my eye or in my skin or my hair or some combination of those 3? And so with these polygenic traits, we're going to see several genes are going to play an important role, and we're going to start to lose our categories here. Another good example of a polygenic trait is something like skin color, which works very, very similar to eye color. Skin color is also due to melanin, and so there's probably at least 3 genes involved here, and they may be similar genes. The melanin gene, how much, and where do you put it? Again, we like to categorize things, including ourselves, so we like to group ourselves, humans do, historically into different races or different ethnic groups based on things like skin color. But again, the reality is if we went out and got 100 people and lined us up from dark to light color, what we would see is it's very hard to draw that line and say, well, here's where one race stops and another race starts. And in fact, I do want to point out that race is very much a social construct, meaning it's part of that need to categorize things, including ourselves. It's not a real biological phenomenon, meaning there is no race gene or an allele that we see that's present in one race and absent in another race. Now, there are a few very minor superficial differences between one race and another in terms of maybe some physical things that we can see, but those are very minor. Most of the genes that we have as humans have to do with things like metabolizing sugars and storing up energy and growing new cells and copying our DNA. And for those types of genes, we're all very similar. And in fact, on average, if I were to compare someone with you, to someone in your own race to someone maybe in a from a distant land from a very different looking race, what we would find is you probably have as many differences genetically with someone in your own race as you would from someone that you would call a different race. And so, again, very much a social issue. There isn't really any big genetic difference at all between different races or different groups of people. One more trait I want to look at here in terms of polygenic traits is height. And height is a nice example because not only is it due to several genes, but we're going to start to see the environment play a much larger role as well. And these are a couple of my favorite pictures, and these are swiped from a different textbook, not your own. But here we see this is a group of students taking biology in 1920, and this is at one of the University of Connecticut extensions. And in 1920, it was an all-male military academy. That's why they're all dressed so nice there. And they're all arranged just by height. And so we've got from short to tall. And again, we can see this continuous variation, kind of a bell-shaped curve. We've got a few short, a few tall, but most people are kind of in the middle. Now, in 1997, they took at the same extension, now it's a co-ed college, and they took all their biology students and lined them up by height again. And so here we can see our continuous variation, again, our bell-shaped curve. Guys are in blue, women are in white. That's the color difference we see there. And the signs are a little bigger, so we can see the height goes there from 5 foot all the way up to 6'5". And the reason these pictures are nice is it really shows us the environmental impact of traits. And today, we have grown up and we live in an environment that's very different than historically and just 100 years ago how people lived. And sometimes I think we lose touch with that because we're familiar with our environment and it's hard to imagine a different environment. Okay? And just as an example, we can see that in 1997, there were 3 guys there that were 6'5". Now, if we look at 1920, the tallest guy in this all-male military academy, so these are kind of the manliest of men in this military academy, was nowhere near 6'5" tall. In fact, the tallest person in this picture was actually only 5'9". And that was the tallest person, right? And so this is a huge difference from 1920 to 1997. Now, obviously, our genes have not changed that much in just a couple generations, and all of a sudden we've got all these tall genes. Instead, what has changed is the environment, especially the environment that kids, our children, are going to undergo as their bones are growing. Okay? Huge improvements in nutrition, having not only more milk, but vitamin D fortified milk, where the calcium and vitamin D allows your body to absorb the calcium into your bones, allows them to grow. Having things like antibiotics. In 1920, penicillin didn't exist as a treatment to treat bacterial diseases. And so we see this very dramatic increase in a very short period of time, really due to this impact of the environment. So kind of keep that in mind, that a lot of things we experience today certainly are very different than the environment our ancestors have experienced in even the relatively recent past, just 3-4 generations ago. Last on our list of exceptions to Mendel's basic ideas are what we would call sex-linked inheritance or sex-linked traits. And these are going to be traits where the gene is located on one of the sex chromosomes, either the X or the Y. And the reason we're going to see differences here with these sex-linked traits is normally, for all of our other chromosomes, remember, men and women both have 2 copies of each chromosome. But for the X chromosome, men are only going to have 1 copy of the X and women have 2. And so if a man were to inherit an X chromosome with the disease allele, for example, that man would have that disease because he only has 1 copy of it. Even if it's recessive, it's going to show up. And so we're going to see X-linked traits, or especially X-linked genetic disorders, are going to be much more common in men than they are in women. And they're always going to show what's called a maternal inheritance pattern. And if we look at the figure here, if we look at a woman's sex chromosomes first, women have 2 copies of the X. One came from mom, one came from dad. But because they're physically the same, we really can't tell by looking which X came from which parent. But because men have different sex chromosomes, an X and a Y, they physically look different. We can tell that Y had to come from dad. Dad was the only one that had a Y chromosome, so the X must have come from mom. And so if there's a disease associated with a gene on the X chromosome, men are going to have it most often, and if a man has it, we know he got that X from his mom, and so that's that maternal inheritance pattern. Mothers passing it on to sons. Now, technically, sex-linked could also include a gene on the Y chromosome. But remember, the Y chromosome is the smallest of all of our chromosomes in humans. Very few genes are associated with it, and most of the genes on the Y chromosome have to do with male reproduction. And so if there was a mutation or a problem with one of the genes on the Y chromosome, the main symptom is sterility, which means that gene would not get passed on, and so it really wouldn't result in any kind of genetic disorder because it wouldn't get passed on from one generation to another. So as we think about sex-linked inheritance, we're really talking about genes on that X chromosome. So let's take a look at how this works. Let's say we've got 2 alleles, a wild type or healthy allele. We'll call that X type A. Okay, X because we're on the X chromosome with our superscript A. And then let's say we've got an allele associated with a genetic disease or disorder, and we'll call that X with a little a because most of these certainly are recessive. So in women, you're going to have 2 copies of the X chromosome. You could be homozygous for type A, or you could be heterozygous, X big A, X little a. And either one of those, you're going to have no problem with the disease. You will not have any symptoms. You will not have the disorder because you've got at least 1 copy of that dominant allele. Although women who are heterozygous, the big A, little a, we would call you a carrier, meaning while you don't have any symptoms of the disease, you have the potential to pass the disease on to the next generation. And certainly many people might be carriers and not be aware even that they're a carrier for disease. The only way for a woman to have these sex-linked disorders then would be to be homozygous for the recessive, to have 2 copies of the X with a little a. But men, since we only have 1 copy of the X, it's going to be different. Men are either going to be X big A Y, which means we're not going to have the disorder, or we may be X little a Y, which means we'll have the disorder. So men cannot be carriers for these sex-linked genes because men don't have a place to hide that second X. Whatever X we get is going to show up because that's the only copy that we're going to have. And again, that's why these traits are often more common in men because it just takes 1 X with little a for a man to have the disease. It takes 2 for a woman to get the same disorder. And by far the most common trait associated with sex-linked inheritance is color blindness. And we've probably all seen these little colorblind charts where you look at the picture and you're supposed to see a number or a letter. I'll be honest, I am colorblind, so when I see these charts, I just see a bunch of little dots there. So hopefully you can see the number or the letter as you look at this chart. It turns out that on average, probably about 1 in every 12 to 20 males here in North America is colorblind one form or another and has trouble reading these kind of charts. It turns out we've got 3 different genes that allow us to see color. 2 of those 3 genes are located on the X chromosome, in particular the red seeing gene and the green seeing gene. And so oftentimes we see red-green color blindness problems that are much more common in men than in women because of this sex-linked issue. And one more exception I want to mention, and this is the rarest of all these different patterns, but it's going to be important because there's a particular trait where this impacts it, and it's probably the trait that I get asked the most about. And this is the idea of what's called sex-influence inheritance. And in sex-influence inheritance, we see that one sex has a trait much more commonly than the other sex. And in this example, men have a trait much more common than women. And when we see that, a little red flag goes up and we think, well, maybe it's sex-linked, maybe it's on the X chromosome. But upon further investigation, we find out that it's not, that it's on one of those autosomal chromosomes numbers 1 through 22. And normally, if that's the case, we would expect both men and women to have the traits equally, since we all have 2 copies of chromosome numbers 1 through 22. So it's kind of conflicting there. We should have equal sex ratio, but we don't. It's out of whack here in one way or another, and so that's why sex is going to influence this inheritance. And what's going on here is a man and a woman may have the same genes, but they may have different phenotypes because the expression of a trait is going to be influenced by sex. And so different phenotypes even with the same genotype depending on what sex you are. And the example here is male pattern baldness. And again, as men, we get bombarded from early childhood We have to look at moms, uncles, nieces, nephews, third cousin, and if they're bald, you're going to be bald when you're older. And we hear all these kind of old wives' tales about how pattern baldness is inherited. Actually, it's a pretty simple pattern, but it's going to have this sex-influenced kind of overlaid on top of it. And so there are two alleles. Let's call H for hair. We'll call H with a big B for the bald allele, and we'll call H with an N for the non-bald allele, or the no pattern baldness allele. Okay? And so if we take a look at genotypes, you could be homozygous for the N or the non-bald allele. And if that's the case, you're not going to have pattern baldness, and it doesn't matter whether you're a woman or a man. 2 copies of HN, no balding problems. Okay. You could be homozygous for the balding allele, HB. And if that's the case, you will have pattern baldness. And again, it doesn't matter whether you're a man or a woman, you'll exhibit this. Now, in our society, it's certainly much less acceptable for women to have that, and so it's covered up much more often in women than it is in men. But it is possible for women to have pattern baldness. Okay. The sex influence aspect comes in. What if you're heterozygous? What happens if you have one of each? In women, that HN, the non-bald allele, is dominant. And so just 1 copy of HN means no balding pattern. But in men, it's just the opposite, that HB is dominant. And having 1 copy of that balding allele is enough to have pattern baldness. And this is why it's much more common in men than in women. Is for men it takes 1 copy, for women it takes 2 to get that pattern baldness. Now, if we think chemically, why would this be? How could the same genes end up being expressed differently in men and women? We have to look at our chemistry. And obviously, chemically, the biggest difference between men and women are our hormones. And so there must be something about the presence of testosterone or the presence of estrogen or progesterone that are influencing this expression and causing the sex-influenced inheritance pattern that we see with pattern baldness. In summary then, to wrap things up, we know that there are many, many traits that do follow Mendel's basic rules or laws, and we would call those Mendelian traits. But as we learn more about genetics, we find that other traits show a much more complex inheritance pattern. And some of the examples we looked at were incomplete dominance, where we have a blending of the phenotypes. Again, our red and white make pink. We could have a multiple allele example, like our blood example, where we've got 3 or more different versions or different alleles. If we look at a particular gene, and we looked at our ABO blood type in humans, certainly as that example. Pleiotropy. We could have one gene affecting several aspects of our phenotype. For example, our hemoglobin, looking at not only sickle cell anemia, but malaria and that heterozygous advantage. Polygenic inheritance, when it takes several genes all working together to give us a particular phenotype, and we looked in this case human examples like height or skin color or eye color, or that pigment in that corn snake was a nice example there. Or we could have sex impacting our inheritance pattern. Sex-linked is the most common pattern associated with that, where we've got genes on the X chromosome. These traits are going to be more common in men, less common in women. And then our last thing we mentioned was that sex-influenced, which is not on the X chromosome, but your sex is going to influence or impact the expression of that particular genotype into a phenotype.